Think about that tiny pill you might take. Seems pretty simple, right?
Deceptively simple, maybe?
Yeah. But what if I told you there's this hidden world of incredible complexity inside, like a really carefully controlled process that's crucial for making sure it actually works?
That's spot on. And right at the heart of creating these life-saving medicines is, well, a key step called crystallization.
Crystallization, okay.
Yeah, the active pharmaceutical ingredients, APIs, those are the bits that actually do the work in a drug. They very often rely on crystallization. And how they crystallize their physical properties, that's absolutely critical.
Critical how? Like for me taking the pill.
Correctly. We're talking about things that impact how that medicine behaves once it's actually inside your body.
Okay, so let's like peel back the label a bit. This deep dive, we're going behind the scenes. Way down to the molecular level to really unpack the science of crystallization.
Right. We want to explore how scientists control this process to make sure the quality is there and ultimately how well the drugs you might depend on actually work.
So our mission today, understand why crystallization is so vital in drug development.
And the pretty fascinating technique scientists use to basically master this whole transformation.
Okay, let's get into it then. Crystallization is not just about, making pretty sparkly crystals you might see in a lab.
Not at all. It's actually a really sophisticated purification step and an engineering step for those APIs.
Engineering. How so?
Think of it like refining and sculpting the active ingredient, getting it into its most effective possible form.
Okay, sculpting the medicine. I like that.
And what's really fascinating is that scientists control several key aspects of this with incredible precision. Three big ones are polymorphism.
Whoa, slow down.
Yeah.
Polymorphism. Sounds technical.
It is a bit, but the concept is key. Polymorphism is basically the ability of one single chemical substance to exist in multiple different crystal structures.
Like different arrangements of the same Lego bricks.
That's a pretty good analogy, actually. Think about it like a key. If the key's got the wrong shapes, the wrong cuts, it won't open the lock, right? Even if it's made of the right metal.
Right.
Polymorphism is kind of like the shape, the internal structure of the drug molecule's crystal. It needs the right structure to work effectively in your body.
Ah, okay, I think I get it. So maybe one crystal form dissolves better than another, or is more stable.
Exactly. Different polymorphs of the exact same drug can have totally different properties. Solubility, how well it dissolves. Stability, how long it lasts without degrading. And bioavailability.
Bioavailability.
Mm-hmm.
That's about how much gets into your system.
Precisely. How much of the drug actually makes it into your bloodstream where it can, do its job. And you can imagine this is absolutely crucial for how well the medicine gets absorbed and ultimately works. We actually see this pop up in research. There was an instance where they struggled because they couldn't develop the right crystallization conditions to separate some very similar molecules, regiosumers. That inability to control the crystal form meant they couldn't isolate the specific molecule they needed. It really highlights how sensitive this process can be.
Wow, so the actual microscopic shape is a huge deal. It's not just having the right chemical.
It's having it in the right structural arrangement. Absolutely. There was a drug candidate, AMG 517, where during development they had to recrystallize it multiple times.
Multiple times. Wow.
To get the specific crystal form they needed, they used first acetone and water, then toluene, different solvents to nudge it into the right structure. It really shows the level of control required. Scientists are essentially selecting the best crystal form for that specific medicine.
That's amazing. Okay, so polymorphism is the internal structure, the shape. What about particle size distribution? That sounds a bit more straightforward.
You'd think so, but particle size is just as critical. The actual size of the API particles and how uniform they are, it has a huge impact.
Like how?
Well, for one, the dissolution rate. How quickly the drug dissolves once you take it. That's directly affected by particle size. Generally, smaller, more uniform particles dissolve faster.
And that means it gets into your system and starts working quicker, right?
Exactly. But it also affects things on the manufacturing side, how well the API powder flows when they're making tablets or filling capsules. Good flow means you get a consistent dose in each one.
Ah, okay. No one wants half a dose.
Definitely not. And for some liquid medicines, like certain soft gels, the particle size needs to be controlled to be incredibly fine. We're talking like 180 micrometers or even smaller.
Which is tiny.
Very tiny, much smaller than a grain of sand. This helps keep the liquid uniform, homogeneous, and helps the drug get absorbed properly, especially if it's a drug that doesn't dissolve easily in water.
Right.
We see this with inhaled medicines, too. Particle size is absolutely vital there to make sure the drug actually reaches the deep parts of the lungs where it needs to work.
So whether it's a pill, a capsule, a liquid, an inhaler, the size of these tiny crystals is carefully engineered.
Engineered is the right word, to make sure you get the right amount of medicine at the right time, in the right place in your body.
Okay. Structure, size.
Yeah.
What's the third key aspect you mentioned?
Purity and yield. Crystallization is actually a fantastic purification technique.
How does it work?
Well, as the desired API molecules arrange themselves into that ordered crystal lattice, they tend to exclude the unwanted stuff. Impurities, byproducts from the synthesis, they often get left behind in the liquid.
So it's like the good molecules find each other and build a crystal, leaving the junk out.
That's a simplified way to put it, but yeah, essentially.
Yeah.
And by carefully optimizing the conditions, the temperature, the solvent, how quickly you cool it, things like that, scientists can dramatically improve the purity.
And yield, that's just how much you get.
Exactly. Yield is the amount of the desired API you actually recover at the end. Optimizing crystallization can significantly boost how much usable drug you get from your starting materials. There are examples in the scientific literature, OP&D papers, for instance, showing really meticulous control of solvents, temperature, purity checks, leading to incredibly high purity, like 99% plus.
Wow. So mastering crystallization isn't just about making the drug work right.
It's also crucial for making sure it's safe, pure, and that you can actually produce enough of it efficiently.
That makes a lot of sense. It's clearly important.
It really is. And controlling this whole process, it's definitely part art, part science. Scientists have a whole toolkit of techniques.
Okay, so how do they control it? What are the tools in that toolkit?
Well, one of the main ones is solvent selection. Choosing the right liquid or mixture of liquids to dissolve the API in.
Why does the solvent matter so much?
Yeah, different solvents dissolve substances differently, obviously. But they also influence how the crystals form, the shape, the size, even which polymorph you get. Finding the right solvent or solvent combination is a really delicate balancing act.
Sounds complex.
It can be. There are even sophisticated model scientists use to try and predict solubility in mixed solvents, like, say, ethanol and 2-propanol mixtures. It shows you the level of detail involved. And you see specific solvent systems mentioned in synthesis procedures, like N-butanoheptane for a particular compound, chosen specifically to get the right crystallization.
So finding the perfect liquid environment is key.
Right.
What else?
Temperature control. Hugely important.
Right, heating and cooling.
Yeah, but it's more than just hot and cold. Temperature affects how much API can dissolve the solubility, but also the kinetics, how fast the crystals actually form and grow. So carefully controlling the temperature profile, maybe cooling slowly or holding at a certain temperature, gives scientists a lot of influence over the final crystal properties.
Okay, solvents, temperature. What else?
Seeding. This is a really clever technique.
Seeding, like planting seeds.
Kind of. You add tiny preformed crystals of the exact polymorph you want into the solution. These are called seed crystals.
And what do they do?
They act like templates or nucleation sites. They basically give the dissolved API molecules a blueprint to follow, encouraging them to crystallize in that same desired structure. It helps ensure you get the right polymorph and can influence particle size too.
Ah, so it's like giving the molecules a little nudge in the right direction.
Exactly, a very controlled nudge. And then there's managing supersaturation.
Supersaturation.
That's basically when you have more API dissolved in the solvent than should normally be stable at that temperature. It's like the solution is overloaded. This overload is the driving force that makes the crystals want to form.
But you need that imbalance.
You need it, but you need to control it carefully. Too much supersaturation too quickly might lead to lots of tiny, poorly formed crystals or the wrong polymorph. Too little, and crystallization might be too slow or not happen at all. So scientists manage how they create and maintain that supersaturation level.
It sounds like juggling a lot of factors.
It is. Even things like the physical equipment matter. The design of the crystallization vessel, how it's mixed, the connections in the plant, especially in pilot plants moving towards large scale, the whole layout for crystallization is carefully considered.
Wow. And how do they even check if they got it right? How do they see these tiny structures?
Well, that's where analytical techniques come in. Very sophisticated ones. We don't need to get into all the jargon, but techniques like solid-state NMR nuclear magnetic resonance can actually tell scientists which polymorphs are present in a sample and even quantify them.
So they have ways to actually see and measure these microscopic details.
Absolutely. They need that feedback to know if the control strategies are working.
Okay. Now, you mentioned gastrosol earlier. Can you connect all this back to a real-world example like that, making a specific drug?
Yeah, the scale-up synthesis of gastrosol is a good case study. Optimizing crystallization is absolutely crucial when you move from making, say, a few grams in a lab flask to producing potentially tons in a massive industrial reactor.
I can only imagine the challenges there. What changes when you scale up?
A lot can change. Maintaining consistent quality is a big one. The yield needs to stay high. The particle properties you achieved in the lab, you need to reproduce those reliably in much larger equipment.
Why is that so hard?
Think about mixing. It's much harder to ensure uniform mixing and temperature throughout a huge tank compared to a small beaker. Heat transfer is different. How you isolate the crystal's filtration, drying that all needs to be scaled up effectively too. Things that weren't an issue before can suddenly become major problems.
So the recipe might need tweaking for the big batch.
Definitely. The crystallization protocol often needs significant optimization during scale-up. We mentioned AMG 517 earlier needing multiple recrystallizations.
Right.
That was partly to get the purity and the specific crystal form needed for large-scale delivery. It shows it can be an iterative process, try something, analyze, adjust, try again until you nail down the conditions for large volumes. And you see this across the board in pharmaceutical development literature. Complex syntheses often rely heavily on a well-optimized crystallization step somewhere along the line to hit the required purity targets.
So figuring out that large-scale crystallization, it sounds like a really critical bottleneck almost, bridging the gap between discovery and actually getting medicine to patients.
It's absolutely a critical control point, and all this effort, this meticulous control over tiny crystal details, it really does have a direct and pretty significant impact on, well, on you, the person potentially taking that medication.
Okay, so connect the dots for me one last time. Why should I, as someone just taking a prescribed pill, really care about polymorphism or particle size?
Because it's the bedrock of that medicine's safety, its efficacy, how well it works, and its consistency. That careful control of polymorphism, it ensures the drug dissolves correctly in your body, gets absorbed properly, does what it's supposed to do,
Okay.
the optimized particle size. That helps guarantee you get the same correct dose every single time you take it, no guesswork.
Right, consistency is key.
And the rigorous purification that comes with well-controlled crystallization. That means you're getting a safe product free from potentially harmful impurities left over from the manufacturing process.
So when I take that pill, I'm really benefiting from all this incredibly precise science and engineering that went into making those tiny crystals just right.
Exactly. It's a testament to the rigor involved. This seemingly microscopic process, crystallization, has a very real, very macroscopic impact on your health and well-being. It truly is one of the unsung heroes in making sure the medicines we trust actually work.
This has been incredibly insightful. So just to sum it up, crystallization isn't just about appearance. It's a vital, super complex process in making APIs, the active part of drugs.
Absolutely.
And scientists use this whole arsenal of techniques, controlling solvents, temperature, seeding, supersaturation, to precisely manage key properties like the crystal structure or polymorphism.
The particle size, the purity, and the overall yield, how much they get.
And getting those crystallization conditions nailed down is just essential for manufacturing drugs safely and effectively, especially on a large scale. It ensures the quality of the medicines we all rely on.
Couldn't have said it better myself.
So maybe the next time you take any medication, just pause for a second. Think about the incredible level of precision, the science, the care that went into creating those tiny crystals inside, making sure they perform exactly as intended. Kind of makes you wonder, doesn't it? What other simple things in our daily lives are hiding that kind of intricate science, those unseen efforts behind the scenes?